Welcome back to Biology B. We're finishing up our first unit on gene expression with the lesson on biotechnology and genetic engineering. Today we'll be exploring the use of gene expression in the scientific advances we've made as a society so far, with a focus on recombinant DNA particularly. Don't forget to check in on your vocab quiz, pulse check, finish the lab we started this week, and if you want to get started on your review guide for the unit test, that'll be extra credit. We're still focusing on the big unit idea about cells and how they're specialized, because even though they have the same DNA and genetic code, they don't always produce the same proteins, which causes them to have different functions. The learning targets for today we're going to focus on is how to explain how DNA technology is used to construct recombinant DNA molecules to alter regular protein production. And we're going to understand how genetic engineering, or biotechnology, is used to produce novel biomedical and agricultural products. Okay, the first thing we have to understand is that when we're talking about bacteria, they don't have the same double helix. They have a double helix, but they don't have the same chromosomal structures that we do as humans. Because bacteria are prokaryotic, which means they're single-celled and they have no nucleus, they don't store their DNA like an organism with a nucleus would. It just freely floats in the cytoplasm. So in this illustration, the red loop is the DNA of the bacteria. The blue circle is the DNA that we're going to use. It's foreign DNA, and I'm going to explain to you throughout the process of this lesson why we would insert that into a bacterial chromosome. So the word for bacterial DNA that we're going to refer to is plasmid. Just understand that bacteria have one chromosome that is a loop. And if we insert a foreign piece of DNA into that loop, because remember, bacteria undergo division asexually, and it's much faster of a process than those of us who have to go through meiosis to reproduce and all of that a multicellular organism involves. So every time a bacterial cell duplicates, it's going to replicate the same loop of foreign DNA included. So when the proteins are made from this bacteria, it's going to make it much faster than we would be able to in our own DNA. But we may have to step back and just remember to compare the difference between prokaryotic and eukaryotic organisms. We learned that last semester. We're going to be hitting it again when we talk about kingdoms in semester 2, but let's start by just comparing what we know about prokaryotic organisms. They have less DNA overall. They're much smaller, much simpler organisms, so there's fewer genes. The chromosome is circular, or like I said, it's a loop. We'll call it a plasmid. And the genes are found closer together than you might find them on our own DNA. Eukaryotic genomes have more DNA. There's more genes as a result. Transcription, or the copying of the DNA to RNA, requires helper proteins like RNA polymerase, for example. The genes are farther apart, and there's a lot of our genome that is not transcribed. They haven't found necessarily genes associated with those stretches of the DNA that aren't copied. So for biotechnology, that's when we use living organisms, like bacteria, to produce materials that we need in bulk because they can make it faster. For example, we're going to be talking about recombinant DNA. If you look at the word recombinant, recombine, so we're combining two different versions of DNA into one molecule, and that we do that because it's more efficient to produce the proteins that we're looking to mass produce. For example, the most common use would be, insulin for one, and the most common host for recombinant DNA is bacterial cells. We use recombinant DNA to make things like vaccines, insulin in bulk form, cancer therapies, asthma treatments. So there's a lot of medical uses for recombinant DNA. All right, bacteria is our number one host when we're creating recombinant DNA, because it's easy to put a gene in bacterial chromosome since it's such a simple loop. Bacteria are very easy to cultivate or grow. They reproduce very quickly, and they produce their own proteins much faster than we do because they have less DNA, less space between, and they have just less to process. So of all of the things that you're going to want to focus on for this lesson, that you're going to notice on your test, the steps for making recombinant DNA will show up on your test. So let's go through these so you understand what's happening. In this first section here, you're taking a gene from a donor. This is a eukaryotic cell. I can tell that it's a eukaryotic cell because there's a nucleus. And what they're going to do is, let's say they want the gene that makes insulin so that we can mass produce it. They're going to remove the gene, just the gene, which is this isolated gene here, from the host DNA. They're going to, with microscopic equipment, of course, surgically insert it into the plasmid of the bacterial cell. So they would literally snip the bacterial chromosome open, and because they have sticky ends, just like everything does, the same DNA in bacteria is the same you'd find in your own eukaryotic cells. So adenine to thymine, cytosine to guanine, the same base pair rules apply. So the ends should seal together, hopefully seamlessly. So this loop, this plasmid with the inserted DNA, that is recombinant DNA. So when I say recombinant DNA molecule, I mean the bacterial, in this case, the bacterial plasmid with the inserted foreign DNA. So the next thing we have to do is reinsert this plasmid with the foreign DNA, this recombinant DNA molecule, back into the bacterial cell, so that when the bacterial process of replication and transcription and translation of its own DNA happens, it's also going to mass produce that one protein from the isolated gene of our host, in this case, the one that will make the insulin. So, let's see, got the plasmid open. So please remember to study these in this order. It's pretty common sense, right? Isolate the donor gene, splice open the plasmid, insert the donor gene, insert the recombinant DNA molecule into the bacteria, and then collect all of the protein that's produced from the bacteria's more rapid transcription and replication or protein synthesis process. Here it is again. Just giving you a minute to screen snip or write down some notes. So, results of recombinant DNA. If a bacterium is inserted with a protein coded by the human genes, it will be added to the bacteria's genome. So it becomes as if it were one piece. The bacteria does not recognize the difference between the bacterial host DNA and the donor DNA, because remember, all DNA, same 4 or 5 nucleotides, right? A to T, C to G, etc. I had said the example we're going to use is insulin. So if you know anybody with insulin, insulin-dependent diabetes, I should say, we need to be able to produce enough insulin to supply all of our population of diabetic individuals. Well, if we have to rely on just slow, regular human transcription and translation to make those proteins, it's going to take a lot longer. So that's why we insert them into bacteria. So again, remember, DNA is a universal code. In other words, adenine, thymine, cytosine, and guanine, the same 4 nucleotides, are in our our DNA and bacterial DNA. And the enzyme RNA polymerase in bacteria will transcribe it no matter whether it's the host or the donor DNA. The thing I want to cover in this lesson is the Human Genome Project. It was a bigger deal back when I was in high school, but if you remember, it's when people with a lot more time on their hands, scientists, have mapped out all of the known genes in our karyotype. So all of the gene locations for the genes that we know about are mapped out, and together that collective is called a genome. It gives us the ability to read the whole blueprint for building a human being. When you compare a human's genome to some other species' genomes, you can see that the genome in bacteria and archaea, which is another form of bacteria, the number of genomes greatly outnumbers the numbers of genes identified in our own genome. So it doesn't really matter how complicated the organism is to determine how many genes there would be. They ask, why do we care? Why do I care about the genome of a fish or raspberries or an apple? And that's because we are using the maps of where organisms' genes are on their genome for genetic engineering, right? How to build a strawberry that stays fresher longer. How do we build an apple that resists decay? All of those processes that we're trying to better to make products that we can benefit from. So again, recombinant DNA is the primary one we've talked about. Positives from genetic engineering, because I know there's some debate about the benefits of genetic engineering, but it can reduce diseases for us. It can provide medications for us, insulin, treatment for hemophilia, treatment to help break down blood clots, anti-cancer drugs, and it can help create new and better crops. This is where a lot of the controversy comes in. But if you have to feed a lot of people and you don't have a great growing environment, you can create crops that grow better in harsh temperatures or lower water areas. We can produce food that's cheaper so that everybody has a chance to eat healthy. We can influence how long they stay fresh during transit so that people can get things like tomatoes that they don't normally have access to, and naturally resisting, bugs and pests. Again, there is debate. We can talk about that when we meet next. Some of the negatives, the more obvious negatives, would be GMOs, right? So genetically modified foods. Some people say they're less nutritious. Some people say they're causing allergies to develop. There's the risk of biological warfare as a result, which sounds, extreme, but is happening. New and more dangerous diseases. They can reduce the genetic diversity in a population. So, for example, when we first started growing corn, when we first settled here, when the Native Americans were growing corn, not all corn looked the same. But since we've sort of streamlined what we're looking for in corn, we've domesticated it, and there's less diversity. Up this lesson, and I want you to go back through and write down any vocabulary terms that you are still not familiar with, and make sure that you go to the Quizlet and study them. I didn't bother putting the links on here because you're not going to be able to click on them, but you should have the link for the Quizlet already. And if you want to input anything into the Padlet, we can talk about that when we meet together next time. So check in with yourself before you finish this recording. How are you feeling about the ability to explain? Would you be able to explain how DNA technology is used to make recombinant DNA molecules and alter the regular protein production? So just to recap, that was this process we're talking about. Would you be comfortable explaining this process, or at least identifying the steps in order? Make sure you can do that. And then, can you understand and explain how biotechnology can produce new and novel benefits and some negatives for us as a population? Okay, that's a wrap. I will see you in the next session. Bye.